Welcome to another deep dive on Fun Facts Online. Today, we are abandoning the macroscopic world entirely. We are going to shrink down to the nanoscopic level to witness a war that happens every single year, often inside your own body. We are talking about the mechanics of influenza. Now, usually when people talk about the flu, they talk about symptoms, fever, aches, fatigue. But that is boring. That is just the aftermath. What we are interested in today is the engineering. We are going to look at the influenza virus not as a bug, but as a machine, a masterclass in biological robotics that performs a sequence of precise, lock-and-key operations to hijack a human cell. This is a story of espionage, shape-shifting, molecular theft, and evolutionary arms races. So let's get right into the anatomy of this shapeshifter. To understand how the invasion works, you have to look at the invader. Influenza A is what we call an enveloped virus. It is essentially a capsule wrapped in a lipid membrane. And this is the first act of theft, because that membrane isn't the virus' own skin. It was stolen. It is a piece of the cell membrane from the last host cell the virus destroyed. But what makes the flu recognizable are the spikes studding this stolen membrane. If you look at it under an electron microscope, it looks like a jagged medieval weapon. These spikes are glycoproteins, and they give the virus its family name, orthomyxoviridae, which comes from the Greek word for mucus. There are two main proteins on the surface that you need to know. Think of them as the hands and the scissors of the virus. The first, and most abundant, is hemagglutinin, or HA. This accounts for about 80% of the surface proteins. It looks a bit like a mushroom. The head of the mushroom is the grappling hook. It binds to receptors on your cells. The stalk of the mushroom hides a spring-loaded weapon we will discuss in a moment. The second protein is neuraminidase, or Na. If HA is the hook used to board the ship, Na is the sword used to cut the ropes when it is time to leave. This is the machinery that defines the flu strains you hear about on the news, like H1N1, or hydrogen nitride. The H stands for hemagglutinin, and the N stands for neuraminidase. But the surface is just the packaging. Inside this lipid envelope, there is a shell made of matrix protein 1, or M1, which gives the virus its rigidity. And piercing through that shell is a tiny, underappreciated pore called the M2 ion channel. Remember that name, M2. It is a proton pump, and it acts as the trigger for the entire invasion. Deep inside the core, we find the blueprint. Unlike many viruses that carry a single long scroll of genetic material, influenza carries a segmented genome. It has 8 separate negative-sense RNA strands. Each strand is wrapped in a protective coat of nucleoprotein and capped with its own copying machine, the RNA polymerase complex. This segmentation is the virus superpower. It allows different flu strains to swap gene segments if they infect the same cell, sort of like shuffling 2 decks of cards together. This is how we get massive pandemics. But let's move to the invasion itself. Part 2. The breach. The invasion begins with a case of mistaken identity. The cells lining your upper respiratory tract are coated in a forest of proteins, and these proteins end in a specific sugar molecule called sialic acid. To your cell, sialic acid is just a standard surface marker. To the influenza virus, it is a landing pad. But here is where the mechanics get incredibly specific. Not all sialic acids are the same. The way the sialic acid connects to the underlying sugar determines everything. In wild birds, like ducks and gulls, the sialic acid is attached via what is called an ALPHA23 linkage. Structurally, this looks like a straight cone. Avian flu viruses have hemagglutinin spikes shaped perfectly to grab this cone. However, in the human upper respiratory tract, the dominant structure is the alpha-2,6 linkage. This looks like a bent umbrella. This molecular geometry is the main reason you don't catch the flu from a pigeon in the park. For a bird flu to jump to humans, it has to mutate the shape of its binding pocket to fit the alpha-2,6 umbrella. We are talking about tiny changes, sometimes just 2 amino acid swaps, like a glutamine to leucine at position 226 and a glycine to serine at position 228. Once those mutations happen, the key fits the lock, and the pandemic potential unlocks. So let's say the virus has the right key. The hemagglutinin binds to the sialic acid on your throat cell. The virus surfs along the surface until the cell, sensing something is attached, decides to swallow it. This is called receptor-mediated endocytosis. The cell membrane wraps around the virus and pinches off, creating a bubble called an endosome. Now the virus is inside the fortress, but it is trapped in a bubble. It needs to get its genetic payload out of that bubble and into the main control room of the cell. To do this, it pulls off one of the most dramatic stunts in structural biology. Part 3. The acid trigger and the harpoon. The cell, thinking it has captured a piece of food or debris, tries to digest it. It starts pumping protons into the endosome to lower the pH, making it acidic. This is exactly what the virus was waiting for. It uses the cell's own defense mechanism as a starting gun. When the pH drops to around 6.0, those M2 ion channels we mentioned earlier flip open. They act as floodgates, allowing the protons to rush inside the virus particle itself. This acid bath inside the virus dissolves the bonds holding the genetic core together, loosening the RNA so it is ready to be released. But the real action happens on the surface. As the pH drops further to about 5.5, the hemagglutinin spikes undergo a massive shapeshift. You have to visualize this. The HA protein is metastable. It is like a loaded spring held back by a safety pin. The acid pulls the pin. In a split second, the protein shoots upward, thrusting a buried hydrophobic sequence called the fusion peptide, literally a molecular harpoon, into the wall of the endosome. It anchors the virus to the bubble wall. Then, the protein collapses back on itself in a jackknife motion, pulling the viral membrane and the endosome membrane together with incredible force. The two membranes merge, a hole opens up, and the viral contents spill out into the cytoplasm. The Trojan horse has opened. Now we enter part 4, the hijack. Most RNA viruses are content to stay in the cytoplasm, the cell's living room. But influenza is an aristocrat. It demands the VIP suite. The viral genetic material travels specifically to the nucleus. It tricks the cell's transport machinery into carrying it through the nuclear pore complex. Once inside, the virus faces a problem. To replicate, it needs to make messenger RNA that the host's ribosomes will read to make protein. But human mRNA has a special chemical cap at the start, a verification code. The virus doesn't have the tools to make this cap, so it steals it. This process is literally called cap-snatching. The viral polymerase binds to a brand new host, mRNA, that the cell is trying to make for itself. It grabs the capped end, and then uses a molecular guillotine to slice the head off the host's message. It then uses that stolen cap as the starter for its own genetic code. This is brutal efficiency. It not only disguises the viral RNA as authentic, but it also sabotages the host cell by destroying its messages, effectively shutting down the cell's ability to call for help. Part 5. The assembly line. The nucleus is now a factory. It pumps out viral mRNA, which goes to the cytoplasm to be built into new spikes and shells. It also pumps out copies of the viral genome. These components have to meet up. The spikes travel through the cell's Golgi apparatus, getting coated in sugars, and are delivered to the cell surface, grouping together in thick patches called lipid rafts. The genetic material flows out of the nucleus to meet them. There is a fascinating mystery here called the packaging problem. Remember, the virus needs exactly one copy of each of the 8 genetic segments to work. If it grabs 2 of segment 3 and misses segment 4, the new virus is a dud. We believe the RNA segments actually form a daisy chain, binding to each other in a specific order to ensure a full set is packaged before the membrane closes around them. Finally, part 6, the escape. The new virus buds off the cell surface, wrapping itself in that lipid raft studded with new spikes. But there is one final trap. The new hemagglutinin spikes immediately try to bind to the sialic acid on the dying cell surface. The virus is tethered to the ship it just sunk. This is where neuraminidase, the sword, comes into play. It acts like a lawnmower. It crawls over the surface, slicing off the sialic acid residues, cutting the tether, and releasing the virus to infect the next cell. This specific moment is actually where drugs like Tamiflu work. They are neuraminidase inhibitors. They gum up the lawnmower blades, leaving the virus stuck to the dying cell, unable to spread. The human body, of course, fights back. We have sensors like RIG-I that detect the alien RNA and trigger interferon alarms, but the flu has a countermeasure, the NS1 protein, a dedicated immune saboteur that hides the viral RNA from detection. Sometimes, the immune system overreacts, flooding the lungs with cytokines, causing the fluid buildup that actually kills people in severe pandemics. It is a perpetual war. The virus drifts, mutating slowly, or it shifts, swapping segments to create monsters we have no immunity against. But by understanding these mechanics, the alpha-2-6 lock, the acid-triggered spring, the cap-snatching heist, we find the targets for our vaccines and our antivirals. It is a terrifying, beautiful, biological machine, and understanding how it works is the only way we stay one step ahead.